guys, we're going to hit complement, and hopefully this won't be too long. So, what I'd like to ask you guys to do first is memorize a phone number for me. It's 1, which is the U.S. area code, 4, 2, 3, 5, 6, 7, 8, and then it's extension 9. Okay? So I'll go ahead and stick this like so. So, and I guess it's actually more like that. There we go. And then that's the US area code. So, why am I having you memorize this phone number? Because this phone number is the classical complement cascade. Okay? So, this is for someone named C. Bab. The C is because these are all C proteins. So it's C1, C4, C2, C3, C5, C6, 7, 8, 9, etc. Bab, B-A-B-B, is because it's 4B, 2A, and then 3B, and then 5B, and all the ones that we aren't using, because each of these is a protein. So I'm going to just put this, let's say that this is C4, okay? And then right now it's inactive because it just is. It's floating in the blood. And when it gets activated, in this case by C1, it will be cleaved into 2 parts, C4A and C4B. Okay? And that's why we have C4A, C4B, C5A, C5B. It's because there's 2 parts, and part of it floats off and part of it stays. This tells you which part stays. Okay? So we can say that C1 and then C4B, C2A, C3B, C5B, and then 6, 7, 8, and 9 are whole proteins. They don't split. So, if you can memorize a phone number, you can memorize complement. So this is, and that's really all there is to it. So 1, 4, 2, 3, and then after that, it's just 5, 6, 7, 8, 9. 1, 4, 2, 3. And if you want...
That used to be the Knoxville area code.
Yeah? Then
and
you've got
lived
it.
in Knoxville. Okay.
Knoxville area code. Nice. I actually was going to look up what area code that was, and I didn't.
Okay, so tell me one more time how the B correlates with the 4 and...
Okay, so we could do it like this. C4B, C2A, C3B, C5B. These are the ones that are attaching to the bacteria. So I'm going to draw the bacteria over here. It's all hairy and it's got its flagella, let's just say. Definitely bacteria, it's got flagella. All right? And these complement proteins, I could put circles around them and then it would be really hard to see what they are, but they're all attached. Okay? And they attach in sequence right there. And this is the classical cascade. Okay? There are 2 other cascades that are actually even simpler than this one. Okay? So without looking, can you tell me that phone number?
1, 4, 2, 3, 5, 6, 7, 8, 9.
Good.
And then C, Bab, B. And
Yep.
they line up with the first 5 numbers.
Good. So you got it. That's the cascade. So next, let's hit the alternative cascade. All right. The alternative cascade literally is just, it starts at the number 3 and it adds 1 new thing. All right. So we have 3B, correct? And it manages to attach directly to the surface just because it hits the bacteria in just the right way. So if you have a C3 protein floating through the blood, the reason it usually doesn't attach is because it hasn't been activated into B yet. So let's actually tell this story and then I'll get to the C3b. So this is floating through the blood, right? And a bunch of them will there's a ton of them in the blood. And so that means the bacteria floating through the blood, hoping it doesn't get killed, is going to be pelleted with a ton of these. Most of them will bounce off, but there is a small region that that is able to attach to the bacteria. And if that region hits with just the right amount of force and just the right direction and just the right everything, then it'll stick. Most things aren't going to have it happen exactly right. That's why most of them don't stick. And I'm kind of simplifying this. There might be another region or something like that too. But there's a region where if it hits with just the right force, just the right turning, just the right everything, then it's going to stick. And that's what makes it happen. Most of them don't have that happen. So they just boing and keep going. They bounce off. And so this one isn't very efficient because most of the stuff hitting it doesn't actually stick. And by the way, just for classical up here, I'll go ahead and stick an antibody because that's often what gets this one started, and then also CRP. So those are some details of what can start it. This one, so if it gets started with a C3 protein, managing to stick and become C3b, because once it sticks, it'll cleave itself in half because the bonds that were keeping it together are now bound to the bacteria, so then it breaks apart. Okay. And C3a will float off, obviously. Anything that's not kept on the bacteria just gets released into the blood. So the other half, like C4a, where is it? It's just in the blood. Okay. C3a, where is it? It's in the blood. All right. So with this second one called the alternative, so I'll put classical up here and alternative down here. Alternative is going to have a protein that is just called B. Literally, by itself, it is the B protein. Why did they call it that? Because they like confusing med students. So, and the B protein floats through the blood just like everything else, and if it sees a C3b attached to a bacteria, then it will stick.
Okay, so that B protein is not the same thing as a B.
It's not C3b, it's not a B cell.
Okay.
It is its own weird little thing, and its whole full name is B. The B protein. Yeah. Okay. Yep, right? So, now I'm going to put it over here again. I'm going to get rid of our little cell that is attached, and we're going to have C3b, and then we have a capital B. That capital B represents the B protein. And then we go ahead and have fun like this. Ditto, 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 ditto. It goes 5, 6, 7, 8, and 9. Okay? It's just the same stuff. As soon as that is turned on, C3b with the B protein, then after this line, it is exactly the same. So, that.
So that 5 would be a 5b?
Yes, it would. It's exactly the same. I'll get rid of all the ditto marks so that it's a little bit clearer. But they just keep piling on exactly the same way. And we can talk about details, but once we have the framework, then we'll go from there.
Okay.
All right, so close your eyes if you'd like to. Tell me, what is the sequence of the classical cascade?
1, 4, 2, 3, 5, 6, 7, 8, 9.
Good. And the alternative cascade, unless there's something else you wanted to do.
And then there's C, bad B that lines up with the first 5 numbers. And then the alternative cascade C3, the little b, and then the big B comes along, and then 5, 6, 7, 8, 9 is exactly the same as in the classical cascade once the big B comes and attaches to C3b.
Nailed it. All right, you've got 2 cascades memorized in less than, let's see, yeah, less than 10 minutes, because you're right at 10 minutes is when you finished reviewing it, so you've got it. All right, now, last cascade. All right, and quickly, also, we'll grab the attachments as well. You already have this attachment. Remember that there's the antibody and the CRP for the attachment. You've got this, the base and the structure. Very good. So the next one is really easy because it is the mannose binding lectin pathway. Mannose is a sugar, and lectin binds sugars, but there are different kinds of lectins. So which kind of lectin are we talking about? We're talking about the lectin that binds to mannose. Instead of saying the lectin that binds to mannose, we just call that lectin mannose binding lectin. So that's why it's called the mannose binding lectin pathway. All right, so we know that mannose binding lectin is going to be a part of this. Mannose associated protein. And so this is just the lectin that we were talking about. And it's called MASP1, and then it can split into a 2. And I'm pretty sure the 1 stays. That's something I should verify. You guys at home, you can pause and verify which one sticks. I'm pretty sure it's MASP1 that sticks. And then after that, you have a C3b, and then our lovely green box. All right, and this is going to directly associate with the bacterial mannose that it's going to express. And actually, usually mannose is on yeast, and so this is the one that's better at yeast. So if you remember that mannose is more common on yeast, then it's like, oh yeah, obviously the one that binds to mannose.
And he's just calling it the lectin pathway.
Lectin pathway is the shortened term for it,
Yeah.
yes. Same thing. So if this is a yeast, then this guy comes down here and binds. But very similar. So these 2 are in bacteria, this bottom one's on yeast. And
fungus?
Fungus, yeah.
Okay.
So
so explain how that one works.
this, well, there's mannose on fungus, you're right, and yeast and fungus, are those separate? I'll just call this fungus to be safe. And so there's mannose on fungus, and so obviously the mannose binding lectin, or MASP1, is going to come down and bind to the fungus. And I don't think he expects us to remember the 1 and the 2 because he didn't touch on it really that much, but it exists. And then the C3b comes after that. And then you have your 5, 6, 7, 8, and 9. All right, so hopefully that helps. I'm going to go ahead and cut it here, and we'll see you guys later.